Imagine a highly specialized structural engineer. Like, they've spent their entire career doing one specific job, which is maintaining the foundation of a massive skyscraper.
Right.
They are absolute experts in load-bearing walls, steel reinforcements, you name it. But then an architectural manager suddenly quits. And without that oversight, this engineer doesn't just, slack off.
Yeah, they don't just take a break.
Exactly. They abruptly drop their tools, they put on a completely different uniform, and they start trying to do the electrical wiring, the plumbing, and the interior design all at once. And they eventually build this chaotic, uncontrolled annex right in the middle of the lobby.
It's a profound identity crisis. And in the microscopic landscape of our biology, that exact phenomenon where a cell just abruptly abandons its established identity to adopt a new erratic one is often the very genesis of aggressive malignancy. We call it lineage reprogramming or stem cell plasticity.
Stem cell plasticity. And capturing that exact transition in real time is basically our mission for this deep dive today. We're pulling from a stack of sources anchored by a genuinely groundbreaking 2025 paper published in Nature Cancer.
Yeah, by Chen Jiang, Yurasong, and their colleagues. It's a fantastic paper.
It really is. They've essentially mapped out this cellular identity crisis, specifically in the context of prostate cancer. And what they found driving it completely flips our standard understanding of oncology on its head.
It really does, because the central premise of this research challenges the conventional linear view of how a tumor initiates. We are so used to thinking about cancer strictly as a disease of runaway proliferation, right?
Like a cell just dividing out of control because of a broken brake pedal.
Exactly. But this paper proves that in many aggressive cancers, before the uncontrolled division really takes off, there is a fundamental transcriptomic rewiring. The cell actively forgets what it is supposed to be.
I want to dig into the mechanics of that forgetting, because we hear the term mutation constantly, but this source leans heavily into lineage plasticity and reprogramming. To ground us, before we look at the data, let's look at the actual architecture of the prostate epithelium.
Yeah, so the architecture is crucial here. The tissue lining the prostate gland is essentially a two-tiered system. You have the basal cells, which sit at the bottom against the basement membrane. They act as the scaffolding.
Like the foundation of the building.
Right, the foundation. And then sitting right above them, you have the luminal cells. Those line the inner ducts and handle all the specialized secretory functions of the prostate.
And in a healthy, mature adult, these lineages are strictly segregated. The paper refers to them as unipotent.
Unipotent, yes.
Meaning, under normal physiological conditions, a basal cell divides to make another basal cell, and a luminal cell divides to make a luminal cell. They stick to their lanes.
They maintain strict lineage fidelity. But, and this is the kicker, embedded deep within the epigenetic landscape of those basal cells is a latent, dormant capability. They are secretly multipotent.
Like our structural engineers secretly knowing how to do all the other jobs in the building.
Exactly. Under severe tissue damage, they actually have the hardwired capacity to generate luminal cells to repair the gland. But that program is heavily locked down under normal circumstances.
So what picks the lock? This is where the paper introduces a classic tumor suppressor gene called Pten. Now, for those listening who follow oncology, Pten is really famous.
Oh, absolutely. It's a master regulator.
It regulates the PI3-AKT signaling pathway, which basically means it keeps cellular survival and growth signals tightly in check. In this study, the researchers utilized a highly specific genetic model, K5-PPN mice.
Right, the mouse model.
They administered a drug called tamoxifen to selectively delete the Pten gene, but only in the basal cells.
And the immediate aftermath of losing that Pten regulation is just staggering. The basal cells don't just start dividing faster. They immediately unlock that dormant multipotency.
They just switch tracks.
Yeah, they begin to shapeshift. They actively transition their transcriptomic profile to become luminal cells.
But the data shows this plasticity isn't uniform, which I found totally fascinating. The researchers mapped this out across the distinct regional zip codes of the mouse prostate.
The different lobes, right?
Yeah, the anterior lobe, the dorsolateral lobe, and the ventral lobe. And the exact same genetic deletion, the exact same loss of Pten behaved wildly differently depending on where the cell physically lived.
All about location.
Right. The shape-shifting was hyper-aggressive in the anterior and dorsolateral regions, driving rapid overgrowth. But it was incredibly sluggish in the ventral region.
Which tells us something profound about the tumor microenvironment, because a mutation doesn't exist in a vacuum. The susceptibility of a cell to undergo lineage reprogramming is heavily dictated by its physical niche. The stromal cells surrounding the epithelium, the local extracellular matrix stiffness, or the baseline signaling environment in the dorsolateral lobe, all of that is fundamentally priming those basal cells to exploit the Pten loss.
So the ventral lobe, for whatever reason, has a microenvironment that suppresses that plasticity.
Exactly.
But I want to challenge that slightly. If Pten's is such a master regulator of the survival pathway, shouldn't its deletion just overwhelm any local microenvironmental signals? Like, why does the zip code matter so much if the main brake pedal is entirely gone?
That is a great question, and it really highlights why plasticity is different from simple proliferation. Pten loss gives a cell a survival advantage, yes. But to physically change its identity from basal to luminal requires a massive epigenetic overhaul.
You mean like opening up the DNA itself?
Yeah, exactly. It requires opening up completely different regions of chromatin to transcribe new genes. The extracellular signals in the dorsolateral lobe, perhaps specific growth factors secreted by the surrounding stroma, are likely providing the necessary cofactors to drive that specific epigenetic remodeling.
Oh, I see.
Without those local cofactors, as we see in the ventral lobe, the cell might proliferate a bit, but it really struggles to fully change its shape.
That makes a lot of sense. The environment supplies the tools for the renovation. So to anchor where we are in the data before we move forward, and to give everyone listening a quick recap,
Always good to summarize.
the prostate relies on a strict two-tier architecture of basal and luminal cells. If you delete the Pten tumor suppressor, the basal cells reactivate a hidden multipotent program and begin shape-shifting into luminal cells. And crucially, this rogue transition is highly dependent on local microenvironments, proving particularly aggressive in the anterior and dorsolateral regions.
Spot on.
Think about your own body for a second, right? How many local zip codes do you have where a disease might act completely differently depending on its exact microscopic location? It's kind of wild to think about.
It absolutely recontextualizes how we think about risk. Now, recognizing that this shape-shifting occurs is one thing. But cells don't just instantly teleport from a basal state to a malignant luminal state. It's a physical, biological transition.
Right, and the researchers really wanted to track that transition frame by frame. To do it, they use single-cell RNA sequencing, or cRNA-seq.
This technology is truly the hero of modern transcriptometry.
It's amazing.
Because instead of taking a bulk tissue sample and grinding it up, which just gives you the average gene expression of millions of cells, like listening to a crowded room as cRNA-seq, isolates individual single cells and sequences their specific messenger RNA.
It's basically a high-resolution cellular audit. You are looking at the precise text messages, the exact instructions being read and executed by a specific cell at a single millisecond in time.
Exactly. Yeah, and by auditing thousands of these individual cells, the researchers used a trajectory inference algorithm called Slingshot.
Slingshot, right.
Yeah, they used it to map out what's known as pseudotachan. They reconstructed the exact chronological pathway of the shapeshifting.
And what the algorithm revealed was a highly specific, reproducible, step-by-step evolutionary bottleneck. The cells must pass through distinct intermediate phases to reach the cancerous state.
They don't just jump straight to the end.
No, they take a journey. Let's walk through that pathway. I like to use the mnemonic BHHP to track the data here. Basal, hilic, hybrid, proximal.
BHHP, it's a great way to remember it.
So the journey starts at B, the normal basal state, which expresses standard basal markers like CAT5 and CART14. Once Ben is deleted, where does the cell go first?
It enters the first H, the hilic-like state. What's fascinating here is that the cell begins strongly expressing CANT13. Yeah, and CANT13 is a keratin protein typically associated with stratified squamous epithelia, like what you find in the esophagus or the tonsils.
Wait, really? So a prostate cell is suddenly producing esophageal structural proteins?
Exactly. It highlights the profound loss of lineage fidelity. The cell is just casting about, accessing gene programs it has absolutely no business using in the prostate.
That's crazy.
And then from this hilic state, it transitions into the second H, which is arguably the most critical finding of the entire paper, the hybrid state.
This hybrid state is incredibly bizarre biologically. The data shows these cells simultaneously expressing CAPORT14, which is the foundational basal marker, alongside AP3.
Right, the aquaporin channel.
Yeah, an aquaporin water channel, typically associated with luminal differentiation and cellular migration. So they are literally straddling two conflicting biological identities at the same time.
Which brings up a really intense biological mystery. Sustaining two conflicting transcriptomic programs simultaneously requires an immense amount of metabolic energy.
It has to be exhausting for the cell.
It is. Normally, a cell caught in a conflicted state like that would trigger apoptosis-programmed cell death. The internal regulatory checkpoints would recognize the fatal error and force the cell to self-destruct.
So what provides the spark to keep it alive? What is the engine actively pushing the cell through this energetically exhausting BHHP trajectory?
That's the million-dollar question.
Because it survives the helic phase, it powers through the hybrid phase, and it eventually settles into the P, the proximal-like luminal state. This proximal state expresses TROP2 and CURT8, and it is the direct launching pad for invasive adenocarcinoma.
Right.
It's kind of like watching a caterpillar turn into a butterfly, but we've paused the video halfway through to study the weird, half-melted cocoon stage. And the key is, this cocoon stage, the hybrid state, is where the cancer initiates.
The malignant butterfly, if you will.
Exactly. Let's do a quick recap here. Using single-cell sequencing and that slingshot algorithm, scientists mapped the exact transition. Basal cells go through helic and hybrid intermediate states before becoming tumor-initiating proximal-like cells. That's the BHHP journey.
Perfect summary.
So if you were a scientist listening to this and trying to stop this process, at which stage of this BHP journey would you deploy your drugs to trap the cell before it turns dangerous? Keep that in mind.
Because that's exactly what the researchers wanted to know. But first, they had to figure out what was fueling the metamorphosis.
Right. What is the engine? And this is where Zhang, Song, and their team deliver a massive paradigm shift. Because when we usually discuss cancer and the immune system, we picture immune cells like T cells and macrophages as the responders, right?
Yeah, and infiltrating the tissue to fight the tumor.
Or we think of chronic extrinsic inflammation like a viral infection or a toxin causing tissue damage that eventually leads to cancer.
Right, inflammation is usually viewed as an external reaction. The cavalry arriving or collateral damage from a war zone.
But the transcriptomic audit of these transitioning cells revealed something completely different. The loss of the Pten gene intrinsically triggered the cell's own innate immune signaling cascades.
Specifically, the NF-kappa B and JAK-STAT pathways were massively upregulated.
So to be precise, this is a cell-autonomous mechanism. The epithelial cell isn't waiting for a macrophage to arrive and cause inflammation. The cell itself is sounding an internal alarm.
Exactly. But the alarm isn't a byproduct of the cancer. It is the architect of the cancer. To prove this, the researchers performed an incredibly elegant experiment using 3D organoids.
Okay, let's break that down. Organoids are essentially miniaturized, simplified organs grown in vitro, right? So they took these prostate epithelial cells and suspended them in a nutrient gel, completely isolating them.
Correct. No blood vessels, no immune cells, no surrounding stroma, just the epithelial cells in a dish.
Totally alone.
And when they induced the Pten deletion in these isolated organoids, the cells still underwent the full shape-shifting process. They formed hyperplastic, overgrown, disease-like structures with filled lumens.
So the internal signaling alone was entirely sufficient to drive the plasticity.
Yes. But here is the clincher. When the researchers genetically deleted P65, which is the primary transcription factor subunit of the NF-kappa B pathway, the lineage plasticity completely halted.
The cells stayed basal. Even without Pten, if you remove the NF-kappa B transcription factor, the cell cannot change its shape.
Precisely. Mechanistically, NF-kappa B isn't just a passive alarm bell. When activated, the P65 protein physically translocates into the cell's nucleus, binds to specific DNA sequences, and actively recruits enzymes to alter the chromatin structure.
It forces the epigenetic unlocking of those luminal and hybrid genes.
Exactly. The internal inflammation is the absolute mechanical requirement for the BHHP transition.
Imagine a worker inside a factory pulling the fire alarm. But instead of putting out a fire, the loud ringing of the alarm actually brainwashes the worker into mutating. The inflammation isn't a reaction to the cancer, it's the architect telling it.
It's a great analogy. The cell isn't just broken, it is actively executing a survival program driven by inflammatory panic.
So just to lock in this concept with another mini recap, because it represents a massive shift in how we view tumor initiation. The loss of Pten turns on the NF-kappa B and JAK-STAT immune pathways inside the cell. This internal inflammation is the direct cause of the cell shapeshifting.
It entirely changes the perspective.
Knowing that inflammation is actually the architect of these rogue cells, how does that change the way you think about chronic inflammation in your own body? It's definitely something to chew on.
Oh, absolutely.
Now, I want to challenge the mechanics of this cell autonomous idea, because the next phase of their research involves pharmacological targeting. If we know the alarm system, we can cut the wires.
Right, the drug trials.
They tested existing drugs on these mice to see if they could halt the plasticity. They used JSH23 to block NF-kappa B, ruxolitinib to block the J kinases, and anakinra to block the IL-1 receptor. And the data shows these drugs successfully froze the cells and prevented tumor initiation.
Which is fantastic news.
It is. But wait, if this entire alarm system is internal and cell autonomous, why would a drug like anakinra work? Anakinra targets the interleukin-1 receptor, which sits on the outside of the cell membrane. Isn't the alarm coming from inside the house?
That is a phenomenal catch, and it speaks to the complexity of the signaling.
Yeah.
You are right that the initial spark is internal. The cornthal loss triggers intracellular stress.
Right.
However, once NF-kappa B is activated internally, it prompts the cell to manufacture and secrete its own inflammatory cytokines, like IL-1, into the immediate extracellular space.
So it creates an autocrine feedback loop.
Exactly. The cell secretes the inflammatory signal and then immediately binds that same signal with its own surface receptors, dramatically amplifying the panic. It's essentially shouting into a megaphone and then listening to its own echo.
Oh, wow.
So when you introduce anakinra to block the external IL-1 receptor, you break that autocrine amplification loop. The initial internal spark might still be there, but it can't sustain the massive transcriptomic overhaul required to reach the hybrid state.
That mechanism is fascinating. And with ruxolitinib, it operates a bit differently, right? It acts intracellularly.
Yes, ruxolitinib acts almost like a molecular wedge. When inflammatory cytokines bind to the surface, they normally trigger the JCAR enzymes inside the cell to phosphorylate, or pass the signal down the chain to the STAT proteins.
Like a bucket brigade.
Right, and ruxolitinib physically lodges itself in the kinase domain of the JAK enzymes, blocking that phosphorylation. The signal never reaches the nucleus, the chromatin never remodels, and the cell is locked in its basal state, unable to complete the BHP journey.
This is incredibly promising data in a K5 Pten mouse model. But we've all seen mouse models fail to translate. The crucial question is, does this specific inflammatory shape-shifting trajectory actually mirror what happens in human patients?
The researchers addressed this directly, and the evidence is really robust. They didn't just rely on cell lines. They analyzed 136 actual human prostate cancer specimens across various clinical stages.
Wow, 130.
Yeah. In human prostate oncology, we use the Gleason scoring system to grade tissue architecture. Gleason groups 4 and 5 represent the most aggressive, dedifferentiated, and high-risk cancers.
The ones most likely to become metastatic and lethal.
Exactly. And when the researchers ran transcriptomic profiling on these high-grade human samples, they found a striking concordance. The exact same molecular signatures that define the mouse hillock and hybrid states were highly enriched in the most aggressive human tumors.
They actually found the BHHP markers.
They found massive upregulation of AQP3, that water channel associated with the hybrid state. They found PIGR, which is a polymeric immunoglobulin receptor deeply tied to the proximal state.
Right.
And crucially, they found intense expression of STAT1, confirming that the JAK-STAT inflammatory pathway was hyperactive in these human tumors.
And if we tie this back to our very first point about zip codes, does the human prostate exhibit the same regional vulnerability as the mouse lobes?
It maps almost perfectly. In human anatomy, approximately 70% of prostate adenocarcinomas arise in what is called the peripheral zone of the gland.
Okay.
When you analyze the baseline transcriptomics of the human peripheral zone, it acts as the biological equivalent to the mouse's dorsolateral, or DLP, region. The local microenvironment in the human peripheral zone inherently possesses the same pro-inflammatory plasticity-enabling signals that made the mouse DLP so vulnerable to Plane's loss.
So the mouse model isn't just an approximation. It's a high-fidelity mirror of the most dangerous forms of human prostate cancer. Let's synthesize this final section before we wrap up.
Sounds good.
We've got existing immune-blocking drugs like ruxolitinib and anakinra that can act as molecular wedges to break this autocrine loop and halt this cancerous shape-shifting in mice. Furthermore, the markers for this process, AQP3, PIGR, STAT1, are strongly linked to the most aggressive Gleason group 4 and 5 prostate cancers in humans.
The very clear connection.
So if we already have drugs that calm these specific immune pathways, what does that mean for the future of preventing aggressive cancers before they even form?
When you look at the totality of this data, it truly forces a paradigm shift in oncology. For decades, our primary strategy has been cytotoxic.
Meaning trying to find and poison cells that are rapidly dividing.
Right. But this research suggests a profound alternative, intercepting the epigenetic signals that tell a cell to abandon its lineage fidelity in the first place.
And that leads us to a highly provocative thought to leave you with. If you are listening to this, you know that chronic inflammation is a hallmark of countless localized tissues, whether from diet, stress, or minor autoimmune activity.
It's everywhere in modern health.
It is. And we've just established that localized autocrine inflammation acts as the direct mechanical fuel for stem cell plasticity and lineage reprogramming. If that's the case, could common targeted anti-inflammatory protocols, perhaps even repurposing existing autoimmune drugs like ruxolitinib or anakinra, one day become the standard prophylactic cancer vaccine for individuals identified as high risk?
It is a really compelling frontier. It suggests that maintaining cellular identity is an active, ongoing process, and that controlling the inflammatory tone of a tissue microenvironment might be just as critical as monitoring genetic mutations.
It fundamentally changes the narrative. It suggests that sometimes preventing a cell from building that rogue factory isn't about attacking the worker. It's simply about cutting the wires to the fire alarm before the panic sets in. Thank you for joining us on this deep dive into the transcriptomic origins of cancer. Take a moment to consider the hidden power of the microenvironments operating within your own biology today.